to the webinar of Amity International Society of Natural Products. On behalf of our Honorable Founder President, Sir Dr. Ashok K. Chauhan, our Chancellor, Sir Dr. Atul Chauhan, Additional Chancellor, Sir Dr. Aseem Chauhan, Vice Chancellor, Ma'am, Dr. Balwinder Shukla, Ma'am, DG AFAF, Dr. Nutan Kaushik, Ma'am, Vice President, Amity Science, Technology and Innovation Foundation Group Captain Kapil Shukla, Sir, the complete scientific fraternity of Amity University, the students and faculty. So I welcome you all to today's webinar. We have a very eminent guest, Dr. Rohidas Arote from, he's an associate professor at Seoul University of he's in North Korea. And
Sorry, South Korea.
sorry, South Korea. Sorry, South Korea.
Don't do not. Don't do not.
I'm so sorry. So he's from Seoul, Republic of Korea, South Korea. And before I invite him to give his webinar, I will just read out the brief resume of his rich experience. So he has done BS in 2000 from University of Pune in pharmacy, and in 2003 he has done his MS in pharmacognosy and phytochemistry from University of Mumbai, India. And in 2009 he did his PhD in biomaterials gene therapy from Seoul National University, Republic of Korea. And presently he is associate professor and also head of the Department of Molecular Genetics, School of Dentistry, Seoul National University, Seoul. And before that, he was research associate at Research Institute of Agriculture and Life Sciences, Seoul National University. And he had been awarded, he had many honors and awards to his credit. He was awarded with Creative Pioneer Research Award in 2017, IAM Scientist Medal in 2017. In 2010, he got Young Scientist Award, Award for Research Excellence from SNU. Then he has also been awarded by Young Scientist Award in 2008 by 16th Polychair, that is World Forum of Advanced Materials. And in 2008 and 9, he was a recipient of Brain Korea Fellowship. In 2000, from 2005 to 8, he was a recipient of Korea Science and Engineering Fellowship. And in 2005-4, he got Best Young Scientist of the Year Award, that is Ajanta Pharma Limited from Mumbai. And he has also cleared his gate by IIT Kharagpur. And his research interests are gene therapy, drug delivery, nanoparticles, biomaterials, cell biology, molecular biology, pharmacology, and toxicology, and theranostics, cancer, immune, and regenerative therapy. He has got 65 publications in peer-reviewed journals, 6 book chapters, and 3 granted patents to his credit. So Professor Rohidas is an associate professor and director of nanomedicine laboratory in the Department of Molecular Genetics, School of Dentistry, Seoul National University. He is one of the leading scientists in the field of biomaterial development for gene delivery, especially for cancer treatment. His research on DNA therapeutics, biodegradable polymeric carriers, and nanoparticles has been published in over 65 international journals and has produced various patents. Professor Arote's lab is focusing on development of novel biomaterials and nanotechnology for a variety of medical applications, including gene drug delivery, diagnosis, bioimaging, and regenerative medicine, also on both fundamental and applied questions in the cross-disciplinary fields of biomaterials and medicine, in order to develop novel therapeutic methods for the treatment of cancer, obesity, and cardiovascular disease. So with this, I welcome you, Professor Arote, a very warm welcome. And I request you to kindly begin your webinar titled Advancing Scientific Research for Future Healthcare, Special Emphasis on Gene Therapy. A very, very warm welcome to you from all of us.
Thank you, Dr. Harsha, for your kind introduction. I also would like to thank all the key members of Amity University for giving me this opportunity to present my research work in this particular well-renowned seminar of Amity. And I'm very happy to get connected to all these Amity family throughout the world on the same time. So thank you very much for the organizers for bringing me on this platform. So I'll be trying to speak mostly about my research work at my research group in Seoul National University, South Korea. I'll just share my screen just a moment. Is it coming now?
Yes, we
Okay.
can see.
Sure. Okay, sure. Okay, so I'll be speaking about mostly on the gene therapy, what we are going to work, and also what we have done till now in case of gene therapy. So we especially focus on the different aspects of the carrier to deliver genes to the target site in case of cancer, in case of different disease. So before going to the specific part, let us have a look of our campus in Seoul National University. This is our medical campus. We have a 4 major colleges: Medical School of Medicine, School of Dentistry, School of Nursing, and also School of Public Health. In this particular campus, then this is our main campus, where we have all these basic sciences, pharmaceuticals, natural products, as well as humanities. So we have like 3 huge campuses. So our School of Dentistry also have a a small existence in this main campus and also in the medical campus. So we have 3 major hospitals, like main hospital, children hospital, dental hospital, and also the cancer hospital. And we are like almost 30,000 students in this particular Seoul National University. There are notable alumni from this university throughout the world, like, all these major sponsors like Samsung, LG, Hyundai, they're all the SNE family people. Well, so coming to the main point, so I'll be speaking about the gene delivery. So as we know that history of gene delivery is something like a long back, since the generation 1 of this conventional drug delivery started. So conventional drug delivery is something like a regular delivery of drugs like tablets, capsules, or liquids, or liquid orals, and some other, which doesn't have kind of a specific key factor to control their release or their efficacy. So these kind of all these conventional therapeutic drugs or delivery systems are called as the generation 1. Then coming to the second one, where we can control the release of the particular drugs, because the drugs which have a small pK value, they get excreted from the body very quickly. So we need to hold them in the body to have particular pK level, so that blood level, so that minimum effective level, so that they can have their therapeutic effect. So in such case, we have to use specific polymers which controls the release of those drugs in a body. So using those polymers, we can also target to the specific tissues, or also a disease site or organ. So in this way, we can control their total movement in the body. So those are coming under the category 2, that is generation 2. And then come next, which is a call as a generation 3, where we actively or selectively deliver genetic materials like DNA, RNA, or mRNA, siRNA, shRNA. At the same time, we also deliver proteins, and also now the recent one is a CRISPR gene editing technology. So those all other comes under this gene delivery system. So gene therapy is nothing but the collection of a kind of a different approaches, and it's particular for the treatment of human diseases, and that's those kind of approaches which rely on the transfer of genetic materials into an individual or animal with help of specific carriers. So this is what gene delivery is all about. Once Professor Inder Verma from Salt Lake Institute, USA, he told, like, gene delivery is nothing but just delivery, and delivery. Because we have a target site, we have a therapeutic gene which is responsible to cure that disease. So how to deliver this gene to the target site, that is what gene delivery is all about. So we have to focus on the delivery ability, because delivering, it doesn't look as simple as what we say. It is very, very a critical task, because once we go in the body, there are several factors which affects the potential of DNA and therapeutic nucleic acids. There are DNase 1 enzyme, so which can easily degrade and kind of a that delivery is very, very important. That is what gene therapy is like we are focusing on right now. So this gene therapy can be used for the cure of different kind of disease, like a cancer or genetic disease, infectious disease, acquired diseases, and so on. So here are kind of like some enlistment, like, this inherited disease as well as the acquired disease. Among these, like cancers or cardiovascular disease or diabetes, neurodegenerative disorders, as well as infectious disease, which are like HIV or AIDS or like other infectious, even corona also. So now also there is a kind of a very upcoming news from USA that the Moderna pharmaceutical, which is from the same group of the people, probably most of the scientists know that Bob Langer from MIT. So Professor Bob Langer, who holds a stake in Moderna Pharmaceutical, now is one of the leading pharmaceutical company from USA in producing or going for the successful for the coronavirus vaccine, which is a mRNA, microRNA based vaccine. So these are all coming up again, like, a DNA therapy, or we call the gene therapy or the DNA vaccine. So that is the mRNA vaccine, I mean, microRNA based vaccine, which is almost in the clinical trial too. So these are the several applications of gene therapy. So if you look at the mechanism of gene therapy, it's very, very simple. As we know, this is a cell. So in case of the cell, human cell or animal cell, we know this is a kind of a plasma membrane. So this plasma membrane is a little bit, I mean, obviously it is hydrophobic in nature, but it also have a kind of a little bit of negative charge on its surface. So what we do, we try to make a complex of this negatively charged or anionic DNA with the cationic polymers. So this will be kind of an anionic ionic interaction. So these polymer DNA complexes, which we call the polyplex. So these polyplexes, which need to be a little bit effectively a positively charged, so that when they come on the cell surface, they need to get adsorbed on this negatively charged surface. And then it will undergo the endocytosis. So this endocytosis, as we know that any foreign material, when it comes in the cell, so endocytosis then is early endosome, then late endosome. And during that particular early endosome and late endosome, whatever the material actually contained in that endosome gets out of that and then comes in the cytosol. But if it doesn't come out of that early or late endosome during specific time, then it goes to the lysosomal degradation and then finally goes to the, goes out of the body. So our main motto is to deliver this nucleic acid material to the cytosol of the cell, where if the nucleic acid is free in the cytosol, then it will be directed towards this nuclear pore membrane, and then it will enter to the nucleus. So once any foreign nucleus once any foreign DNA or genetic materials comes in the nucleus, the protein synthesizing assembly will give kind of a signal, okay, a new gene has come. So based on the sequence, let us start the synthesis of the protein of that particular gene. So DNA to RNA, RNA to protein, and then further on. So this is a kind of a simple mechanism of gene therapy. So we are trying to modulate, or we are trying to deliver this particular DNA to the target site. So there are four major elements of gene therapy. So first is, obviously we know this targeted disease, like what disease we're going to use these genes, and which genes are responsible for that. So this is the work of the biotechnology and the molecular biology people, like who works mostly, okay, this particular disease, okay, this gene will be responsible for what kind of cancer. So, and once the gene is delivered, then the gene will express, it will show its effect. So that gene expression will be in the form of the proteins, or like upregulation or downregulation. So the main part of my research laboratory is to synthesize and develop this gene carrier. So we have a target disease, we know the therapeutic gene. So how to deliver that gene to the target site, that is the gene carrier. So main work of our laboratory is focusing on that. So until now, there are like various types of gene carriers been developed. Gene carrier is also called as a gene vector or vector. So there are like viral vectors, and also there are non-viral vectors. So viral vectors are all like viruses, retroviruses, lentiviruses, adeno-associated viruses, adenoviruses, herb simplex viruses. There are several viruses, baculoviruses, even there are some kind of insect viruses also is being used. So these are all the several viruses are being used as the carriers to deliver the gene. But the problem with these viruses is something like very, very strange, that they are immunogenic. And once this gene is been delivered to the target site, these viral particles undergo the self-replication. So that is another very, very big issue. Also, sometimes, like if you want to deliver a big, large size of the DNA to the cell, then these small viral particles cannot carry that. Or sometimes, even if they carry, there is an issue of this insertional mutagenesis. So these are all the several side effects. And also there were some kind of negative effects. I mean, negative results have also been found during the clinical trial, that there were the death of some patients during the clinical trial. So that's the reason actually it's very, very important to ensure the safety of these carriers in order to use those materials for the human purpose. So looking at this particular disadvantages of viral vectors, so various scientists, they switch their focus from viral vectors to non-viral vectors. So among the non-viral vectors, all these polymers, lipids, nanoparticles, and the combination of polymers and lipids, which are called as the polyplexes. Sometimes people also use sonoporation, electroporation, or gene gun. So these are several kind of techniques. So these all comes under this non-viral category. So among those, the polymers are the one where we have a kind of a versatile platform where we can tune the properties of the polymer depending on the pathophysiology of disease, and we can target specific moiety so that we can achieve a targeting. So these are the kind of properties which we can get through this polymer or even the liposome. So my laboratory is focusing mostly on that, and we try to achieve a similar therapeutic effect or even higher therapeutic effect by synthesizing those non-toxic polymers. So until now, if you can see, there are different protocols, clinical trials by specific vectors. And I would say viruses obviously the tops the list, because viruses are very popular to deliver a gene, but they were very toxic as well. But some people thought, okay, let us think about what our work has been done. So let us focus on the work has been done. So let us not think about mostly on the toxic effect. But if we are applying for the human being, obviously toxicity is very, very important, and we have to ensure this safety. So retroviruses, adenoviruses, lipofection, and all the other different kind of viruses have been used maximally for the maximum clinical trial, then followed by these lipid particles, then naked DNA, and so on, and like polymers. So if you can look at another very, very interesting data, like which countries are leading in this particular clinical trials, especially using the gene therapy. And as we know that the US always tops the list with several clinical trials going on. This is in fact a little bit old data, but the recent data shows like there are plenty of clinical trials, especially on the cancer going on, especially on the immunotherapy, then followed by China, Japan, India, and also even the Spain, South Korea, and so many countries they're doing clinical trials. Also, among different types of diseases like cancer or neurological disease or musculoskeletal diseases or cardiovascular. So all the diseases are the target of cancer diseases are the target of gene therapy, but cancer is one of the major leading death in the world, and that is actually one of the major target of the gene therapy as well. So there are plenty of clinical trials going with this cancer. There are millions of pathways going on in cancer, starting from the glioblastoma from the neurons to from that till the toes of our leg. So there are so many different types of cancers available in our body, and all different cancers have their own different pathophysiology, different pathways to get kind of a malignancy. And so there are like several key points where we can control those particular pathways using gene therapy. Then these are the cell types which are majorly being targeted in case of gene therapy. And like, for example, we have adipose tissues, we have epithelial tissues, we have a fibroblast, we have immune cells, we have a mesenchymal stem cells. So immunocells are one of the major cells which are which have been setting a platform for the gene therapy. So until now, several clinical trials, especially in the cancer, have been completed around these immunocells. As we know that some 3-4 years back, a Nobel Prize was given to a person, especially in physiology. That particular Nobel Prize was given to a immunotherapy person, because this is kind of one of the hot issue and leading one of the field in case of the molecular biology and medicine. So this particular immunotherapy is one of the very, very leading field where several different leading scientists are working on this immunocells and immunotherapy. Then followed by this mesenchymal stem cells, then comes the bone marrow cells or adipose cells and simple fibroblasts as well as simple epithelial cells. So immunocells are the major target of the gene therapy. These are the different companies which are involved in the production of development of different medicines through gene therapy, like Oxford Biomedica or Gene Enzyme or Transgene, Targeted Genetics. And they also follow different ways, like some medicines are based on the retroviruses, some are based on the non-viral such as polymers or lipids, Some are based on adenoviruses. So these are the different companies that have been involved in production of gene therapeutic materials. So we know that there are different kind of barriers in the cell, and these barriers are like one of the rate limiting steps. So any foreign material, or specifically a genetic material, if we deliver in the body, there are different kind of barriers, like extracellular barriers. Firstly, like if you give it by IP or IV, so blood, I mean RBCs or WBCs or serum proteins, they are the one of the barrier, because these particular barriers or proteins, they interact with these genetic materials and they diminish their activity. Or sometimes, like a carrier, for example, we use the cationic polymers. So cationic polymers, along with the cationic polymers, we deliver genetic material like DNA or even RNA. But when it comes in the contact with the serum proteins, they form the aggregate, and ultimately they lead to the toxicity. Also, sometimes the tightly packed endothelial cells inside the blood capillaries, they also kind of restrict the delivery of these nucleic acid materials. So this normal vasculature as well as the tumor vasculature, both we have to think in very detail in order to develop therapeutic systems using the polymers. So once this genetic material, it goes in the cell, that is that intracellular delivery. So in case inside the cell, there are like, firstly is obviously the plasma membrane, which is endosome. So endosome is acidic in nature. So whatever the foreign material or nanoparticles actually gets adsorbed on the cell membrane, then it will be endocytosed, and that endosome will have acidic nature. So this is like what we call this early endosome, then comes the late endosome, and then during this particular time, early endosome to late endosome, so that particular 1 or 2 hours, if the material of that endosome or contents gets released in the cytosol, that is very good system. But sometimes some materials doesn't come out of that. So as I said, like it goes to the late endosome and then finally to the lysosome and then degradation. So it goes to the body without showing any action. So we are trying to employ such materials which try to eject this particular material or cargo from the endosome to the cytosol. So we are using specific polymers which contain these amine functionalities, and these functionalities polymers are like they have a high buffering capacity. So because of that buffering capacity and also a proton sponge effect, these materials come out of the body, I mean especially come out of the endosome and then gets released in the cytosol. So there are different kind of materials we have to use in order to control these extracellular barriers as well as intracellular barriers. For example, in case of the extracellular barriers, we have to use some kind of inert materials which controls the targeting and aggregation with the different serum proteins and the blood materials, and also the targeting moieties for the specific transportation of complex in the blood. Because as we know, in case of the extracellular barrier, there is also a reticular endothelial system. So that system also kind of cleanses the body very quickly. So our material should not be cleansed easily throughout the body. Also, once the material gets inside the cell, the first, as I said, the first membrane is the cell membrane, then comes the endosomal membrane, then the nuclear membrane. So these all have to be successfully crossed by our system. And also the DNA releasing at right time, at the right place, also is very one of the rate limiting step. Otherwise, there is no use of our material. It will go without any unaffected, and it will go showing without any kind of a therapeutic effect. So we require kind of a cationic groups for successful gene delivery, and that balance is also very important. So as I was explaining about the various clinical trials, so this is kind of a timeline of gene therapy. So in this particular timeline, starting from September 1990 until today, till date, there are several clinical trials being done on this gene therapy space, especially in case of the MD Anderson Center in USA and several other universities. So among all these successful clinical trials, these two big blocks, black boxes, that is June 1999 and October 2002, these are only the two kind of a negative, these are only the two negative results in case of the gene therapy, because where there was a death of a patient during the clinical trial. And since the viruses were being used as a vector or as a carrier, and those viruses kind of shows their unexpected results, self-replication, and also some other immunogenic effect. That's the reason there was a death of these two patients in these two particular clinical trials. Apart from that, since September 1990, there was the first clinical trial was successfully done for the treatment of this French Anderson patient with severe combined immunodeficiency syndrome, SCID, which the severe combined severe combined immunodeficiency syndrome is something where this adenosine deaminase is one enzyme which is lacking in this particular patient. And that patient is not able to stay in the normal environment with normal condition. So these are kind of a very, very immune deficiency syndrome. So that was successfully treated by gene therapy. Then followed by this June 2000, there was another completion of the draft sequence of the human genome. So based on that, there were so many other clinical trials were done. There is also a very, very good story about the bubble boy. So this bubble boy, as I said, like, you know, because of the one particular missing gene in his body, he was not able to get survive in a normal environment in the world. So he was being kept in the bubble with particular conditions. So that was being successfully treated by gene therapy as well. So there was like several positive effects, positive sides of gene therapy. So whatever system we develop, we have to consider these three major considerations. We have to develop and focus specifically, like specificity. So only the desired cell should be targeted, not all the cell, because in order to achieve the specificity and targeting, then we have to focus on specific properties of the materials and the pathophysiology of disease. So once we consider and ensure those things, the specificity can be easy. Then followed by the efficacy. Even at the small doses, our material should be potent enough to target and to cure the disease. So that efficacy is important, as well as a safety, which is very, very important. Third one, that because any material we develop should be safe, non-toxic, non-immunogenic, non-inflammatory, and also the tumor generation. Should be non-tumor generative. So in this way, like, you know, whatever the materials we are making, these systems should be safe enough so that we can get a FDA approval, and then we can go further. So coming to the research part, in our laboratory, we are using the polyethylenimine based polymers. So this polyethylenimine is one of the very popularly used polymers, especially in case of the paper pulp industry. But it has a very good application in gene delivery because of its buffering capacity and nucleophilicity. So since it has like three types of amine groups, like primary, secondary, and tertiary, it has a high buffering capacity as well. And also it has a proton sponge effect, because this was firstly reported by J.P. Behr from France in 1995. So the paper was published in Proceedings of National Academy of Sciences long back, 1995. And he reported first time that this polymer is one of the golden standard polymer, because it has very high transfection efficiency. It is very effective in all different kind of routes of administration, like in vitro, in vivo, specifically like intramuscular, intravenous, intraperitoneal, intratumoral, and so on. Even aerosol also is very, very effective. But the problem with this polymer is that it is toxic. So because it has, it is a non-biodegradable. It doesn't get degradable in physiological pH. So because of that high charge capacity and a high nucleophilicity, it also targets other non-target cells, and also get kind of interacted with the blood components, serum proteins, and it leads to toxicity. Another is a non-biodegradability. So the major issue is like the toxicity of this particular polymer. So it can carry genes on effective pace, on effective way, but it is very, very toxic. So my laboratory decided to use this polyethylenimine, which is a low molecular weight. Because if you use the high molecular weight, it has a high capacity to deliver gene. That is, we call the transfection efficiency. So the problem is not being solved. So we have to use a small molecular weight, low molecular weight polyethylenimine. Because why we are using amine functional group? Because amine functionalities are very, very essential to have a condensation, better condensation with phosphoric acid of DNA. Because DNA need to be, it's like a spiral structure, which is a polymer in nature. So it has a, like, all this coil structure has to be like properly condensed and compacted in one nanoparticle. So in order to have that, we need to have a long chain polymers which have a high buffering capacity, as well as we should have a amine functionality in our polymer chain, so that the polymer of the amine of the polymer to the phosphate of the DNA, they have a very good high affinity, so that they can have a very nice ionic complex at physiological level, and that ionic complex is tight enough to protect the DNA as well. So we decided to synthesize a library of a hyperbranch polyester amines, which are like we denote as a PEA, using the low molecular weight polyethylenimine, and different kinds of monomers like a polycaprolactone, or glycerol, or sorbitol, or lactitol, or xylitol, or mannitol. So those are kind of different kinds of sugar alcohol-based monomers and their acrylates. So that this particular hyperbranch polyestyramine should be water soluble, because whatever the material we develop, it should be water soluble at physiological level, physiological pH and physiological solution, because it has to go well in the body. So water solubility is important. It also have a better DNA condensation because of the presence of amine functionalities. It will also have a high buffering capacity because of the presence of this polyethyleneamine, so that it will have a better endosomal escape because of the proton sponge effect. It will have a low toxicity because we are trying to employ this ester linkage in our backbone. So that ester linkages are very susceptible for hydrolysis at physiological levels, which will lead to, after hydrolysis, it will lead to a production of acid and alcohol, right, of that particular material. So these ester linkages, like amide linkages as well as disulfide linkages, so these are all like susceptible and biodegradable linkages at physiological level. So our first success was to synthesize this polycaprolactone with polyethyleneamine. So here we, as I said, like polyethyleneamine, if you use the high molecular weight polyethyleneamine, PEI, it is a very toxic and it has high transfection efficiency also. It has a proton sponge effect as well. So we are trying to employ the low molecular weight polyethyleneamine. And as we know that polycaprolactone is a US FDA approved molecule. It is a non-toxic, hydrophobic. It is also a biodegradable one. So the resulting polyestyramine, which will synthesize by copolymerization of this polycaprolactone and PI, will be biodegradable, high transfection efficient. It will have a low toxicity. So this is our main objective, main hypothesis. So we decided to synthesize this polycaprolactone diacrylate. This is the polycaprolactone diacrylate. We can see the vinyl groups at both the ends here. To this polyethyleneamine, which is a branch polyethyleneamine. Here is the primary amine group, tertiary and secondary here. So by Michael addition, we reacted this particular two materials at anaddress level using 40 degree as a temperature, 40 degrees Celsius. So this is a representative structure of this polyestyramine where we can see the ester linkages. So these ester linkages are very susceptible for the hydrolysis, and they are like gives kind of a boost at non-toxic polymer. So this particular material was very, very effective and successful to deliver gene. So we have checked its toxicity also. I am not showing the toxicity data here. So even at high concentration, there was no toxicity signs found in kind of a 293 or HG2 or HeLa cell lines, any cell lines. This is a kidney carcinoma cell line. HG2 is a hepatic carcinoma cell line, and HeLa is cervical carcinoma cell line. So in all these three cell lines, we checked the transfection efficiency by luciferase assay. So here we have used PGL3 as luciferase as a reporter gene. We know that luciferase gene kind of exhibit particular chemiluminescence. So that chemiluminescence will be measured by a chemiluminemeter, and that we call as RLU, that is relative light units per milligram of the protein. So those particular RLU lights were plotted against the positive control. Here we have used this pink bar. You can see this PEI 25 kilodalton. So this is a positive control we have used here, which is high transfection efficient polymer. But compared with that, our polymer, synthesized polymer, like this dark blue or light blue bars, they were even much better. So these are because of the low toxicity of the polymer. We also compared this transfection efficiencies with another positive control, that is a lipofectamine. In all the three data, we can see that synthesized polymer is having a much better transfection efficiency compared with the PI 25 kilodalton, which is a positive control. We also changed the reporter gene. From luciferase gene, we shifted to the GFP, which is a green fluorescence protein, and we checked that in all the NP ratios like 10, 20, even 20 and 30 and high NP ratios, the PI toxicity, because of the PI toxicity, the transfection efficiency is reduced. But in case of the polymer, which is a PCL PI 1.8, we can see this light bar, it has a high transfection efficiency. Also, we decided to deliver these materials by aerosol administration. So in our Seoul National University School of Veterinary Medicine Hospital, we have developed very, very unique system that is called NOEC chamber. That is like nose only exposure chamber. So in one pipe, they have designed in such a way that we can put 6 different mice in 6 by making the holes, and then we can aspirate the material and aerosolize. So we can, there are like minimal losses. And once the inhalation of this GFP gene using our polymer, then 2 days exposure time was given. And then after the sacrifice of the mice, we dissected and we found that the fluorescence. So this is a kind of a fluorescence. We can see the difference. This is the PI 25 kilodalton, which is a positive control. It's the PCL PI, which is our carrier. So it shows a high fluorescence compared with the positive control. So this indicates the advantage of our polymer over the positive control. Then next, further after getting this success, we decided to go for the another sugar alcohol based molecule, that is a glycerol. So glycerol is like very hydrophilic, as we all know. It's a non-toxic, hydrophilic, and biodegradable. And another parameter is that it has a high osmotic, hyperosmotic effect, because it is osmotically active. Because of that particular property, the the cellular uptake of this particular material is very, very easy. So that is also one of the key points we kept in mind in order to develop this particular polymer. So it was co-polymerized with polyethyleneamine, and then effective polyestyramine was synthesized by again, this Michael addition reaction, which is very, very simple at 60 degrees Celsius with anhydrous methanol. So these polyestyramines based on glycerol dimethacrylate. Another one is here glycerol triacrylate, which is, this is again kind of a hyperbranch structure. Because of the triacrylate, there are 3 acrylate groups. So wherever there is a vinyl groups, so whenever there is a vinyl groups, those particular vinyl groups are get kind of added to these amine functionalities with the help of this Michael addition reaction. So these are actually, we have to take in specifically in mind that these particular materials are being used successfully to in vitro to deliver genes. So we have kind of a different cell lines we have used, like cancer cell lines or normal cell lines in vitro as well as in vivo, and by we check the transfection efficiency also and the toxicity as well. All data was excellent in case of Luciferase assay, as well as using the green fluorescence protein by the flow cytometry and different cells. So this indicates the advantage of our polymer compared with the positive control, such as this green bar, which is a PI 25, and also the lipofectamine as another marketed positive control. So this indicates kind of advantage. Also, we check in the similar way the aerosol administration, by aerosol administration, the gene expression pattern, and it was found that glycerol-based polymer shows high transfection efficiency. You can see the high fluorescence here compared with the control and the PI 25 kilodalton. These are the kind of merge images, like phase contrast and the fluorescent both. So in this way, we went on synthesizing and developing different kinds of materials which are non-toxic, biodegradable, and also specific. But here in particular cases, we haven't achieved any kind of a specificity. These are all kind of a non-specific, because when we deliver these nanoparticles in the systemic circulation, they keep rotating near the cells, either healthy or even the target, the disease cells. But they sometimes doesn't go even contact to the disease cells, because we haven't kind of given any kind of a specific targeting moiety to these nanoparticles. So later we thought, like, let us go further to have a specific conjunction or conjugation of different kind of a specific moieties, such as folic acid. As we know that most of the tumors, they overexpress folate receptors. So if you conjugate the folic acid moiety to our polymer, the folate interaction will lead to the high uptake of our nanoparticles in the cell. And in this way, we can achieve our high therapeutic effect. So in this particular, another for successful polymer, which is a lactitol-based polymer. In this lactitol-based polymer, there are 4 or 5 free hydroxyl groups. So it is very highly hydrophilic polymer. So what we did with the help of acrylic chloride, we modified this polymer to the lactitol diacrylate. And then this lactitol diacrylate polymer was added to this branch polyethyleneamine, which is 1200 Dalton molecular weight, by Michael addition reaction in the DMSO, anhydrous DMSO. And this is a representative structure of this polylactitol-based transporter. So we have given the name PLT, polylactitol transporter, PLT. So this PLT vector or PLT polymer is having a combination of this caveolé uptake part, which has a free hydroxyl group for the uptake by the caveolé-mediated endocytosis at the cellular surface. Then the second part is the degradable part, which is a ester linkage part, and the third part is amine functional part, which is responsible for the DNA binding. So these are the 3 key points actually we incorporated in one system, and then we make the complexes with the DNA. And after the making the complexes with polymer DNA, this which is called the PLT DNA complexes. So these complexes were treated for the transfection efficiency in different cell lines. And it was found that you can see here the data that transfection efficiency, you know, as we go on increasing the dose, like 20, 30, and even more, this red bar of the PI DNA was getting reduced and slowly coming down, but the blue bar was still consistent and showing high transfection efficiency. So this indicates another advantage of another new polymer. So because since this polylactitol-based polymer, it is kind of a sugar alcohol. So we decided to go for pancreatic cancer. As we know that the parenchymatous cells of the pancreas, they overexpress these asialoglycoprotein receptors, which are kind of a sugar receptors we call. So these sugar receptors have affinity, high affinity for these sugar alcohol-based polymers or even the polysaccharides. So since this is a sugar alcohol-based polymer, so we decided to use this polymer for specifically for the hepatic cancer. So what we did, we have no idea like whether it will have a same affinity for all the organs or only specific affinity for liver. So what we did, we labeled this DNA by Cy 5.5 as a molecular probe. And then after making the complex with our polylactitol system, PLT polymer, after that, these complexes were delivered by intraperitoneal administration to the mice, And after that, we check the fluorescence in the different organs. And it was found that liver was one of the highly targeted organ by this system. So this indicates that this, our polymer-based system have a high affinity for the liver. I mean, parenchymatous cells of the liver, because parenchymatous cells of the liver, they overexpress this asialoglycoprotein receptor, which are the sugar receptors. And our sugar alcohol-based polymer has a high affinity for that. So that is kind of a receptor-mediated endocytosis. So this is kind of one of the key factor. Another polymer is like sorbitol-based polymer. So this sorbital dimethacrylate and linear PI also in the similar way synthesize this sorbitol-based part, which has a high free hydroxyl groups are responsible for hyperosmotic effect and high uptake. This particular ester linkage part is responsible for the degradation, and the amine functionality part is for the endosomal escape and as well as the targeting with the DNAs. So this polysorbitol-based system also in the similar way, we conjugated with we coupled with made the complex of the DNA and then delivered. But in this case, we did another one specific thing that we wanted to join the folic acid. So this folic acid, as I said, this folic acid is hydrophobic in nature. So initially we activated the folic acid. After the activation, this activated folic acid, we conjugated with bifunctional polyethylene glycol. And this bifunctional So this bifunctional polyethylene glycol, we conjugated with activated folic acid, and then this folate PG was again added to our polymer, which shows this polysorbitol co-PI folate, this particular system. So in this folic acid part, which is at this particular end, is responsible for a folate receptor targeting, this ester linkage, gene binding part, osmotic stimulating part. So these are all like having their own functionalities. So in this way, we can have a specific achievement and specific targeting of these particular properties in the one system itself. So after the conjugation with DNA, we have delivered this material to the cells. But here we decided to go for not DNA, but siRNA. So as we know that small interfering RNAs have a high potential, high therapeutic potential in several diseases. In case of the DNA has to go in the nucleus. In case of the DNA has to go to the nucleus so that it can have a DNA to RNA and RNA to protein, and then mRNA will come out and then it will synthesize its own responsible protein. But in case of the siRNA, it is a kind of a cytosolic phenomena. So siRNA has to act in the cytosol. So we have a one step reduce, one step less compared to the DNA. So this siRNA of the OPA1 protein, OPA1 is like optic atrophy protein, and it is responsible for the mitochondrial fusion of any cell. So if you control this mitochondrial fusion during the cell doubling, so we can control the cell growth, and in this way we can achieve the apoptosis of the cancer cell. And that was the key point we kept in mind. So we decided we gave the sequence to the company. We got the SI-OPA1. This is the siRNA of the OPA1 protein, and we combined with made complexes of the polymers with this SI-OPA1, and delivered, and we found that we can have a reduced gene expression. We can see this particular protein expression here in FPS, which is now folate conjugated polyestyramine here. So this FPS shows a less number of or less intense OPA1 expression, indicating that it achieved the apoptosis. It kills the cell, or it kind of paralyze the mitochondrial function. So in this way, we can achieve apoptosis in cancer cells. We also check this particular by the tumor model of the mouse. Here we can see this particular biopsy mice. I mean, this is a kind of a xenograft mice model. So in this xenograft mice model, it is a control. This is a scramble. Scramble is like, it has the same sequence of siRNA, but it is a non-therapeutic. So it doesn't have actually actual effect. And this is actually SIOPA1. So we can see the number of reduced number of tumors compared with the control and the scramble. This indicates a polymer system is very, very effective. And after conjugating with this therapeutic SIOPA1, which is a very, very essential siRNA to control the apoptosis of the cancer, it shows the less number of tumors. We also confirm by another Western blot analysis using this expression of protein, OPA1 proteins, and also the immunohistochemical stainings. So this all data was excellent, and it was one of the, I mean, we filed a patent for this some years back. After that, another previous polymer, as I said, like, polycaprolactone PC, I mean, that is PCLPI. So that polymer also, we decided to conjugate the folic acid because we had to achieve a specificity, specific targeting. Otherwise, if you go a non-specific way, it takes a lot of efforts as such, and we get a very minimal positive effects. So we use a polyethylene glycol as a linker here. So as we know that PEG is one of the non-toxic and hydrophilic material which is approved by the FDA, and it engages the solubility as well as the stability in the systemic circulation. Most of the pharmaceutical scientists knows that this PEG, we call the stealth technology, is one of the popular, and PEG is one of the very popular material in formulations. So this particular PG kind of decreases all these immune reactions and prolongs this circulation time. So this is one of the key points we kept in mind, and we use here as a bifunctional PG. So in one side it is acidic, one side is amine functional group, and then we have polyethylene glycol chain. This PG chain we decided to use initially 5,000 Dalton molecular weight, but then later we go for a lower one because if you go on increasing the molecular weight of polyethylene glycol, the transfection efficiency probably sometimes goes down. So we have to optimize in such a way that it should not be very high, it should not be very small, but in a moderate enough to achieve that our desired effect. So we, as I earlier experiment, we have activated the folic acid. This is the folic acid structure by DCC NHS chemistry. After the activation, we use bifunctional PG. This is a PG molecule with amine functionalities at one side and carboxylic at one side. So this bifunctional PG was added in the basic pH using pyridine. So this is a folate PG structure. So here we have a amide bond here of this folate PG. So this folate PG was by then again added to our final polymer, which is a PEA, polyestyramine, that polycaprolactone polyethylene polymer, PEA. So this is a polyethylene part, and this is the polycaprolactone backbone. So this PEA polymer was added by simple reaction EDC and NHS, and then we can see the structure of this particular folate PEG PCLPI. So this folate is responsible as again for targeting the folic acid moieties and ester linkages for degradation. So by NMR we could found that almost 11.2 mole% of folic acid was conjugated to the final polymer, because minimum 5% is sufficient to target the folate receptors. So we check its toxicity in different cell lines at different concentrations like 10, 20, 30, 40, 60 micrograms per ml. And we also check the concentration of polyethylene glycol, polyethylene imine 25 kilodalton, because it is a positive control. So we also need to check how toxic it is. And at 60 microgram per ml concentration, we can see hardly 30% of the cell viability is available for PI. But in our polymer, we can see almost 70%. So this indicates like our polymer is non-toxic at all the concentrations compared with PI 25 kilodalton. So we check the transfection efficiency, especially the receptor-mediated endocytosis. So since our polymer contains the folic acid moiety, so we have to check the folate receptor containing cancer cells and folate receptors, I mean non-folate receptor containing cells. So we know here that HepG2 and HepG2 and KB. KB is a mouth carcinoma cell line and HepG2 is a hepatic carcinoma cell line. So in case of the KB carcinoma cell line, shows abundant number of folate receptors. So since we applied this material to the KB cell line, we can see the higher transfection efficiency compared with the HepG2 because it doesn't have any folate receptors. So we can see that our carrier actually follows this receptor-mediated endocytosis of the KB cell lines. We also in another way also we check like whether our polymer really follows this receptor-mediated endocytosis or not. So what we did, we kind of pre-block folate receptors by using 200 nanomole of the free folic acid. So it was kind of a pre-treatment given to the cell before the transfection. So we have used the 200 nanomole of the folic acid, external folic acid. We pre-treated the cells. So we can see that it's a red and green bar, which is like without folic acid and with folic acid. So with folic acid are the green bars. So in this with folic acid, we have actually blocked the folate receptors by external folic acid, and then transfected the cells. So since the folate receptors were already blocked by the folic acid, so there were no more scope for the polymers to actually follow that pathway and to enter the cells and show the effect. So that's the reason actually we got a less transfection efficiency here compared with this without folic acid. So in this way, we made a different kind of studies where we can see that our polymer really followed this receptor-mediated endocytosis pathway in the KB cells. So now currently we are working on these two polymers, which is like diurethane dimethacrylate, which we name as a UD, and triethylene glycol dimethacrylate. This is called as a TG. So this UD and TG polymers are another very unique polymers. These are synthesis of the Michael addition reaction simple way, because we want to keep the synthesis scheme as simple as possible, because it should be industrially feasible if it comes and if it needed, because long and 3, 4, 5 step reactions always looks better on the paper, but industrially they are not feasible. So we have to see all the cost effective way as well, and also industrial feasibility also. So this microaddition is one of the very effective way to synthesize such polymers. So this is the structure of UDPI polymer, and this is the structure of this TGPI polymer. Here we use branch PI by Michael addition reaction. So both the polymers were characterized by NMR. I'm not showing the data here. NMR, IR. Also, we check the particle size by the photal and also zeta potential, because zeta potential is also very, very important parameter in case of the gene delivery, because zeta potential, the final charge is very, very essential to know for the people. Then we check the transfection efficiency by MTS assay in 293 cell, HeLa cell, HepG2, and also this A549 cell. In all the cell lines, we can see here, UDTG have a high cell viability, but this green bar, which is of the PI 25 kilodalton, is very low, and that is because of the toxicity. So this positive control, it's a positive control, but it shows high toxicity, but our polymers doesn't. So this indicates the safety of the polymer at in vitro level. So once we achieve the 60% cell viability, we can go for the in vivo experiment and animal studies. We also check the transfection. Transfection were fantastic as well in all the cell lines. So I'll just go a little bit further here. Another one is very, very important, that bapillomycin effect. As I said, that polyethylene-based polymers, they have a proton sponge effect. And then proton sponge effect is very, very important parameter of only the PI-based polymers. So for that, we check this bapillomycin effect, because bapillomycin is like a vacuolar type of proton ATPase inhibitor. So once we block these receptors, the proton sponge doesn't work anymore. So after blocking these particular receptors with and without bapillomycin, we can see here the transfection efficiency was different. That means that once the pretreatment of these particular receptors, vacuolar type of receptors by bapillomycin, there was no scope for the polymer to go and show this proton sponge effect. So in this way, we compared and we got kind of advantage that whenever we apply this system, it shows that particular parameter. We also check, when we give these materials by systemic circulation, there is a serum, right, in blood serum. So sometimes serum protein also interact with the materials and they kind of create toxicity. So we should also ensure that our system is kind of sufficient and capable enough to be delivered by systemic circulation, such as IV or IM on different way. So we should check the interaction of serum or the serum effect. So with and without serum, you can see here, without serum and with serum, as such, no much profound difference was found in the transfection efficiency. We use the 10% of the fetal bovine serum here, and we use the A549 cell line, and there was no much difference. So this indicates that even if like our system being delivered by a systemic circulation, there won't be any kind of a toxic effect in the presence of serum. Now, this is a very important parameter. Here right now we are working on the microglial polarization because microglias are kind of immune cells of a brain, I mean immune cells of nervous system, and they have a high role in case of the nerve regeneration, apoptosis, as well as neurogenesis, and also a repair, because this particular microglia, and along with the astrocyte, they have a very important physiological functions like pro-inflammatory properties, like damage or cell death, or even the curing the neuropathic pain. So those particular parameters or diseases are in order to set up or in order to work on those particular treatment of those diseases like spinal cord injury or neuropathic pain, we have to use this microglia as well as astrocyte in a very effective way. In case of microglia, they have a very unique properties, because once we activate the microglia, they undergo this M1 and M2 phase, and these both the phases are very essential. So we decided to activate the microglia by delivering a specific material. So we use our material like TG. So this TG, that's the previous polymer, and we conjugated this material by Alexa 488 as another molecular probe, and we just check it like after the intrathecal administration at the cervical area of the mice. After giving this particular injection, I mean delivering this polymer Alexa 448 material, we checked the expressions, and we found that most of the, I mean, cervical region or even thoracic or lumbar sacral, most of the times the microglia were targeted, because since our target is mostly the microglia, but microglia are not separate cells. They are like mixed with the astrocytes and along with other neuronal cells. So we have to target only the microglia. So it is very important to achieve that target-oriented effect. We checked in different region the injection site, and then we found that in case of cervical, thoracic, and even lumbar or sacral, we found that microglia are one of the most targeted cells in this intrathecal administration way. Even neuron also was targeted here. So we used two concentrations of material, 1 microgram and 5 microgram, but then 5 microgram doesn't have as such profound effect. So we just kept on the one itself. So now later, what we decided to use DNA. First, before that, we check the toxicity. In case of the primary cell line, because microglia are very unique cells, and to make those primary cells of the microglia is very tough task. So our students were working hard to make these primary cell lines of microglia, and then we check the cell viability of our polymers in microglia primary cells. You can see the data here. In case of the MTS primary UD and TG, both the polymers, they showed very high cell viability compared with PI 25 kilodalton as a positive control, even at high concentrations such as NP ratio 30 and 40. So this kind of data indicates that polymer was non-toxic even in the primary microglia cell. And whenever we do an experiment on primary microglia, which are as equal as the animal study, since these are the same microglia derived from the animal. So we decided to go further and to check the transfection efficiency on another way. We check it here, and we can see here, in case of the primary mixed glia cell, the transfection efficiency was higher in case of UD polymer and TG polymer as well in the second figure. So this primary microglia, it means that almost we are getting a good result in case of the animal study, in case of a transfection efficiency. In another cell line also we check here, that was also fantastic. So this indicates that this particular TGP polymer or even UD polymer with the help of pDNA, which will lead to this particular copolymerization and then this nanoplex. So this nanoplex was delivered by intrathecal administration. In some way, we also use the intracranial. And then we decided to go it further for a better way. Well, I'll just escape this part, cell viability, okay, fine. So it was found that, we also wanted to check the uptake kinetic. So we complex this DNA, reporter DNA, reporter genes like PGL3 luciferase with Alexa 546 and Alexa 488. So these are the different markers, biomarkers of the microglia. And at different time points, like 5 hours and 24 hours, we check the uptake kinetics. So PGL3 Alexa 546 is the red colored dye. We can see here this particular red dots, which shows the uptake of the nanoparticles after 5 hours and 24 hours. In the same way, the green fluorescence after 5 hours and 24 hours. So this indicates that polymers are being, polymer and this PGL3, LX546 and this material are being uptaken along with. So in case of this dye, if we replace the dye with the gene, it will be also uptaken in the similar way. That is the model. So we also check the uptake kinetics and the transfection efficiency of Alexa-488 nanoplexes in the primary glial cells here. We can, in the figure B, we can see this particular transfection efficiency, this mean fluorescence and the NP ratio in 5, 10, 20, 30, all the NP ratios we can see this transfection efficiency compared with to the CD11 and ACS Assay, these two particular biomarkers. So these, all the data shows the positivity of our carrier in a very effective way. This is also another system where we use the spinal cord regional delivery of the nanoplexes by the intraspinal administration. And by the flow cytometrical analysis, we found that almost the CD11 expression was almost higher. This indicates when we use our system polymer compared with the positive control that have a high affinity towards this astrocyte and not, sorry, microglia and not the astrocyte. This is also another brain microglia specific delivery by the intracranial administration, because since it is in brain, so we use the intracranial administration in the hippocampus region, and we found that the highest expression of the CD11 B plus cofactor. So in this way, I would like to conclude. I think I took a little bit more time. So I just want to conclude our data in such a way that we have, successfully synthesized by Microglia reaction all this library, huge library of the polymers. We characterize well physicochemically as well as spectroscopically, like using the NMR, IR, or even all other like nanoparticle parameters such as particle size or zeta potential or morphology by same time. I didn't show the data here, but they were been successful done. Also, these polymers were having a biodegradable linkage, so they were non-toxic. They have high transfection efficiency, and they also keeps kind of one other promise to have a kind of a tuning versatile platform for another new polymer system. We can also couple different kind of a targeting moieties such as folic acid or transferrin or integrin or different kind of a vitamin B6 and so on. Also, we can target or conjugate these cell penetrating peptides, CPPs, for better efficacy. So these all polymers having show they have a high transfection efficiency and all were non-toxic. Also feasibly, very feasible for animal studies, and also for having a small size nanoparticles which are less than 200 nanometer. That is a good application for the intracellular delivery. So this is the one. These are all our collaborators for this work as well, and our previous work from the international and national level. Also the industrial collaborators from Korea, Samil Pharmaceuticals as well. So this is our group who done most of the work, and these are the two PhD students actually who work mostly this part as well. And there were so many other people who are behind this work. So I would like to thank you all for your attention, and I'll be happy to answer your questions if you have any. Thank you.
Thank you so much, Dr. Rohidas. Before I request our esteemed panelists to share their views, I take their permission to ask a few questions, 2-3 questions, because there are around 9 to 10 questions. And later on, maybe, I'll share your mail ID with everyone so that after you answer, they can, with your permission only, I'll share your mail ID.
Always welcome,
Yeah. So the first question is from Bhagyashri Saharia. One is from Mubashir Gyani. He's asking why cancer treatment is so expensive. Will gene therapy play a role in reduction of the cost?
It is. A lot of clinical trials are ongoing, but as we all know that we have several millions of pathways in our body, especially for the cancer. Like if any tumor grows, so there is not only a one way it grows. There are different other factors responsible for that pathophysiology and also mutagenesis as well as the malignancy. So if you try to control one, obviously there are 10 other, by which it gets consumes a lot of energy and food materials and it grows. So it is very difficult for us. Like it is like the situation in Corona in India right now. So
Yeah.
we have so many different measures, but still Corona is not able to control. It is not in our hands. So it's similar happening with the cancer as well. So that just 2 days before there was a death of Professor Sam Gambhir from
Yeah, I
Stanford
saw in
University.
your
It
LinkedIn.
is
Yeah.
such a sad news because he's one of the leading scientists in case of the radiology. He used to detect, he is the person who is expert in detecting the cancer, but somehow he got
Yeah.
that cancer. So it is very, very complicated disease, and it is not like in our hand, but we are trying our level best to give a therapeutic effect and translation medicines in a different way. Yeah.
Yeah. The next question is from Bhagyashree Saharia. She's asking, sir, how gene therapy is applicable to epigenetic disorders, and also what are the best known applicable softwares for drug design?
Well, in case of the drug design, I don't work in more than that, but since our work is about the delivery of genetic materials. So in case of epigenetics, yes, the gene therapy has several applications, as I explained in my, I think, 9th or 10th slide, that in the timeline that, it is used in all different types of genetic disorders. Earlier, even we can cure several genetic disorders using the gene therapy. There was an example of severe combined immunodeficiency syndrome. So that is particularly a genetic one. So that is because of the deficiency of this adenosine deaminase enzyme.
Yeah.
When the bubble boy enzyme, if you can Google out just like a bubble boy, you can get all the details about how bubble boy was cured. So these are all so epigenetic is all the hereditary disease also can be cured by gene therapy, and also the acquired disease as well, cardiovascular, or even the we can use a germline gene therapy as well as a somatic gene therapy. So there are plenty of applications in gene therapy. The only issue is that we have to ensure a safety in case of a carrier, because we know the gene here, we have a disease here, but how to deliver the gene to the disease site, that is one of the major tasks for all the scientists. And that is what we are working on. So yes, there are plenty of applications of gene therapy.
Okay. And this is from an anonymous attendee. He has asked many questions, so I'll just take one of his.
Okay.
Like, this long chain polymer is successfully, transferred gene by endocytosis mode. But how does it pass through nuclear membrane?
Oh, polymer has not to pass to nuclear membrane because there
Yeah.
is no work of polymer in nucleus.
Yes.
Only the polymer is used as a carrier.
Carrier, yeah.
So it has to go only until the cytosol. So once it's got in cytosol, the DNA will be separated, and then DNA will go to the nucleus.
Yeah.
So there is no polymer.
So I request others to please mail him. I'll be sharing the mail in the Amity Herbal and Natural Product group, both in Facebook and LinkedIn. And then you can pose directly questions to Dr. Rohit Das. And now I request our esteemed panelist, Group Captain Kapil Shukla sir and Rajiv Sharma sir, who have joined us. Rajiv Sharma sir is actually a scientific advisor to our Honorable Founder President sir. And he's also he was ex secretary serb and was also executive director for Indo-US relations. And as I told you, sir is vice president for Amity Science, Technology and Innovation Foundation, Group Captain Kapil Shukla.
Yeah.
So I request both of them to kindly share their thoughts. Yeah.
Oh, Dr. Shukla, I think we are in Facebook, so we know very well. Okay.
Captain Kapil Shukla sir. Okay, you know.
You're right.
Yes, sir.
Wonderful, like Dr. Rohidas.
Thank you, sir.
You have given really wonderful talk, and you are doing really great work. I think gene therapy is perhaps the future.
Yes, sir. Thank you, sir.
Holds a lot of potential for benefit of mankind, and you actually explained it in such a easy way, actually. Otherwise, I know it's a slightly complex subject, but very, very easy. You have actually explained this thing. So again,
Thank you, sir.
Thank you very much, actually, and I'd like to congratulate Dr. Harsha Kharakwal to convince you to give a talk here, actually. So it was really great to listen to you, hear to you. Yeah. Thank you.
Yes, sir. It's absolutely pleasure to be with you, sir. I mean, I don't know, I didn't expect you will join, but that's very good of you. Like you joined.
Yeah, and actually, I was in SNU basically 2010.
Oh, really? Then
Yeah, I
okay.
actually accompanied our Minister of Science and Technology, Dr. Mr. Bansal was our minister.
Oh, yes, yes, yes. I
Yeah,
was
so that
okay,
time I
okay.
visited SNU actually that.
Oh, I didn't know that actually. I would have hosted you then. I didn't know that.
But 10 years back, okay. So,
Dr. Rohitas sir, in his WhatsApp picture he has with Modi ji, all of them,
Oh,
I think
yeah.
it's in Korea, hai na?
yeah. I mean, last year when he visited South Korea, so we Indians group hosted Prime Minister that time. Yes, yes.
So now I
So
request Group Captain Kapil Shukla sir also.
yes, sir. Yes, Shukla sir.
Thank you so much, Dr. Rohidas, for a very informative lecture presentation, and it was.
Okay.
Thank you. I think there is some voice disturbances.
Voice
Yeah.
breakage.
Yes,
So
network some.
network problem.
Yes,
So
yes.
actually, everyone, if you see, there are more than 50 chats. Actually, few of them I have answered. So all of them have really appreciated your lecture. And
Can you hear me?
yes, sir.
Yes,
Now
sir.
we
You're
can
back,
hear
sir.
you.
Yes, sir. We can hear you.
Thank you, Dr. Rohidas. Sorry, there's some connectivity problem because of the rays.
Yes, sir.
Yes, sir.
Thank you, sir. Thank you, sir. My
So
pleasure.
I must thank you very much on behalf of the Amity University, on founder president's behalf, as well as the chancellor's behalf.
Thank you, sir. Thank you very much, sir. My pleasure.
So, Dr. Rohidas, as sir told, as Rajiv Sharma sir told, like this is the future, and we also look forward for collaborating with you, both for projects and for student exchange and faculty exchange program.
Oh, yes, yes. That will be very good platform and very good initiative in such way, because I actually spent almost 15 years in South Korea, and I mean, we have learned expertise, got expertise in such field, but I really would like to bring such kind of techniques to our India, because
Yes,
we
yes.
have an immense potential in our India and a lot of talent.
Hmm.
So given the facility and particular infrastructure, they will do
Yeah.
blast in such case.
Because Dr. Rajiv Sharma sir is also Director General for Amity International Alliances. So sir looks after the complete international part of research.
Okay.
So
Yes.
we look forward for such collaborations, and like, so on behalf of Honorable Founder President Sir, Chancellor Sir, Vice Chancellor Ma'am, and the complete scientific fraternity. Sir, would you like to say anything more, sir, before we conclude? Rajiv Sharma sir?
Yeah, actually, of course, we have actually very good collaboration with Korea. Research actually. So, and biotechnology, I think, is a subject of collaboration there actually.
Yeah,
Yes,
bio is very strong in Korea.
Yeah, so whenever actually this call comes, I'm sure actually Amity University and with you actually we can develop some good proposals.
Oh, yeah, that will be wonderful, sir. I mean, it will be a pleasure to work with you and connect with you in different ways. In fact, we have recently established one ARN, Asia Research Network. So through that, with one professor from Hanyang University, he's in charge. So we are kind of trying to connect the Indian universities and make it so. In that movement, we propose to Indian government and Korean government to establish one kind of like, a research institute, India-Korea Research Institute for both the countries. And that was been approved by Department of Science and Technology when the last year Korean president came to India, I mean, 2 years back. So this is something like probably if it goes in proper way, and then hopefully that will be one of the good bridge of the scientific collaboration between two countries.
Yes, yes. So surely, so thank you so much, Dr. Rohit Das, and thank you all the participants for patiently, waiting, and thank you so much, Dr. Rohit Das. Everyone was highly benefited, and everyone is asking for. So you can go to Amity. People are asking for a link. I'll just share with them.
Sure.
Like, we have Amity Herbal and Natural Product Group both in Facebook and in LinkedIn. So those of you all who are working in that allied areas can join that group and get benefited, and we'll be sharing the lecture also very soon with all of you. So
Sure, my pleasure.
Yeah.
So thank you very much, all the organizers, Dr. Saurabh, Dr. Harsha, and I'm really happy to see Dr. Rajiv Sharma sir also and also Shukla sir to listen my talk. So I'm really overwhelmed. So thank you very much giving me this chance and opportunity to present my research work, what we are doing abroad abroad and showing it in India. So this is something kind of one of the good platforms. So thank you for giving this nice webinar platform for all the scientists.
Yeah.
Thank you, sir.
So
Thank you, madam.
thank you, everyone. Have a great evening. Thank you, sir.
Thank you, sir.
Thank you, everyone. Okay.
Thank
good.
you. Thank you.
Good.